Doctors Longo and Roberts discussed the current status of gene therapies in rare neuromuscular disorders in this 8-part podcast series. This is derived from the symposium that was presented at World Symposium 2025 in San Diego, California on February 4 through 7, 2025, and is intended for healthcare professionals only. This podcast includes information about investigational compounds that do not yet have a regulatory approval or authorization for a specific indication. The safety and efficacy of the agents under investigation have not been established, and contents of this podcast shall not be used in any manner to directly or indirectly promote or sell the product for unapproved uses. The views, thoughts, and opinions expressed in this presentation belong solely to the author and are subject to change without notice. The contents of this presentation do not constitute an endorsement of any product or indication by Astellas. In this part, Dr. Longo will discuss gene replacement therapy in lysosomal disorders.
Now let's go back a second to gene therapy. Gene therapy obviously has the potential of answering many of the questions that we still have open in lysosomal disorder, because they could restore the activity of the lysosome pretty much in the whole body, or at least in multiple tissue. And as you have seen, gene therapy can be done ex vivo, where we take cells from the affected patient, we correct the gene, or we put an extra gene that it is functional, and then we put them back by doing a bone marrow transplant, basically creating space for the cells that have been genetically modified to correct the lysosomal defect. And then the biggest approach, this is done usually by lentiviruses that they integrate inside the genome. The second type of approach is with in vivo gene therapy, where it is given systemically, and in most cases, this is done utilizing an adeno-associated virus, and there are different types that have affinity for different tissue that allow them to direct them where the gene is actually necessary. And obviously that allows you to target the delivery of the gene. In addition, by using specific promoter, you try to restrict expression of the gene in the tissue that need them. And this is very important because, for example, if you express this gene in the dendritic cells that are involved in the immune reaction, you have an increased chance of having a bad reaction to the gene therapy. So you want the gene therapy to be restricted to the tissue and the organ that needs them. And finally, there are more and more examples of in vivo administration restricted to certain tissue. Obviously, delivery to the CNS is important because many gene therapy do not cross the blood-brain barrier, even though some of the newest ones seem to have access to it as well, even when given systemically. There are also examples of administration of gene therapy directly inside the eye, or directly inside the cochlea to correct, in the last case, some type of hearing loss. And, and some of these approaches seem to be already relatively successful.
In the next part, Dr. Longo will discuss ongoing gene therapies in lysosomal disorders.